Okay, we're recording.
Right.
So, one of the things that I think we miss in class, because we either have a video by one of us that nobody watches, or in my case, I do a whole lecture and people say, 10 minutes, 45 minutes, I'm not going to watch that. And then we have some conversation in class, several days later, is you miss out on the discussion. So Bob and I are going to discuss some things. And
We do every week.
yeah, and one of them is about precision and accuracy. And we just finally got a paper published after a year, almost a year of fighting with a hostile reviewer and a semi-hostile editor. But and actually a year is not my record. My record's 5 years. Bob, I don't know what yours is.
15.
15? Okay, you win.
Issue engineering, yeah. 15. It was a long and hard road, but I ended up, yeah. There are times when things don't work out with papers, and you've got to wonder whether or not it's was an editorial decision to kill something, or it just incompetence, just a bad system.
Yeah.
Lots of things, yeah.
Yeah. And, one of the things that you told me before, and I think would be apropos to hear about now, is I know that you sort of irritated somebody by calibrating the machinery in your lab when you were a grad student or postdoc, and your then mentor, all of a sudden, all his papers didn't work out so well.
Yeah, that was a wonderful event. So I was a new graduate student, and had learned a little bit about calibration and how important that is. And many of you may not know about that. It's if you haven't you might have done this in a physics lab, you probably have in a chemistry lab somewhere, but calibration is really essential. And if you don't calibrate your instruments, your measuring devices against something that's known, a lot of people do calibration completely wrong. They'll calibrate the same data against the same data. They think that, I did it twice, it's calibrated, like it's repeated. But nope, I had calibrated our measuring devices for muscle physiology, actually, when I was in graduate school. And all of a sudden, the effect that my advisor had been publishing for more than 10 years, it was almost 15 years, it just vanished. And it was because they had been measuring something wrong. It happens with alarming frequency. And so a postdoc in my laboratory, very smart guy named Neil Cole, I was just starting as a graduate student. He was already a postdoc, so he had many years more experience than I did. And he said, this kind of stuff happens all the time. We started talking about it, and he's like, I'm going to go through and look at some of these old papers, these old important papers in muscle physiology. And he collected a bunch of them, and then he and I reviewed them for correctness to see whether or not the paper was actually done correctly using what information they give you in the paper. Did they make the measurement correctly? Where we couldn't even calibrate their equipment, but we could say, as an engineer, that's not an accurate measurement. Turned out that 85% of the papers were not using the correctly calibrated or even just the correct type of instrument. 85%. So it's very common in science, as Mark and I have discovered to our horror, that it's very common in science that measurements are simply done incorrectly by people. And it's basically your job. It's probably front and foremost your job as a biomedical engineer. And two types of people: biomedical engineers and biophysicists should know the difference. You should know what you're measuring. And a big part of that comes out to be the scientific instrument. And then part of that is, the difference between accuracy and precision. What accuracy, how accurate is your measurement over the entire range that you're measuring. In my advisor's case, he was clipping the data. So it turns out that his amplifier was only working up to 6.2 volts. And so even though he kept turning it up, it was never increasing the signal. So one of the groups where he was trying to show, had a very different stimulus threshold. He just was never able to reach it. He thought when he dialed it to 7 or 8 or 9 volts that it was at 7 or 8 or 9, but it was always plateaued at 6.2. And so he was able to show huge differences physiologically between tissue from older animals versus tissue from younger animals. But that difference turned out not to exist. There was just a stimulus threshold. There wasn't a physio. There was the threshold was slightly above. Turned out his plateau. The difference wasn't really there, but his bad instrumentation made it appear that he was getting these huge measurements, right? Which he wasn't getting. You know, if he could have gone to 6.25, he would have, and the two sets of data would have been very close, but he kept turning the dial up, oh, these 10 volts or 12 volts or whatever. And no. So yeah, he didn't know how to use his instrument. And in the field of muscle physiology, which he dominated at that time, in terms of numbers of papers, you know, you say, okay, well, this is an outlier. It only happened in this one case where one guy was using the wrong instrument. No way. This is a phenomenon that's happening across science. Every time Mark and I look at something with, you know, carefully.
And it's more widespread than you think, and most people watching this may even be guilty of some of this. My first job in the neurophysiology lab that I started out in when I was the in-house engineer for a neurophysiology lab. My first thing, my first and foremost, most important job was calibrate everything for every experiment. In other words, make sure when we said we had 1/10 of a millisecond accuracy for collecting neural data, we actually had that. And so I would go through a long process and check everything and make sure all the timing of all the stimulators, all the recording devices, everything was working. And, you know, that was the most important thing. And so anytime I approached an experiment, you had to look at and decide, well, is this being recorded correctly? And the very first time we switched over, well, you know, at the time we were using an LSI 1120, which for nobody listening to this will know what that is, but you do, Bob, because that was a precursor to the PC. I mean, that was the, you know, and the very first PC was 10 times faster than that. And your phone is how many thousands of times faster than that. But the very first time I tried to use a PC with Windows, all of a sudden I had 30 millisecond delays popping up all over the place. And it drove me crazy. And then, you know, when we started, you know, building out the brain gauge and doing reaction time testing, we did the same thing. Well, let's make sure we're accurate. And so what we did was we did a robotics test. Using robotics, we actually tested the brain gauge, and then we tested all the methods that everybody else in the world is using. So most people, for reaction time, they flash something on the screen, and then they hit a key or they hit the mouse. And, you know, that is not accurate. So, and that's what the paper was about.
Yeah, so the paper really points out something that, and we come right out and say it. I mean, you know, the modern computers have a core that's very fast, you know, gigahertz, right? But that doesn't mean that it's measuring time for events on human interface devices in, you know, the inverse of that frequency. That does not mean that at all. But a lot of people, and Mark and I have both heard this more times than I could count. Oh, well, computers are faster now, so they're more accurate.
Yeah, I mean, we even got that comment on one of the homeworks. Somebody read an old paper from like 1870s doing reaction time, and they assumed that today's computers were faster, and that is and more accurate. And that there were no computers in the 1870s, or not used in those experiments, to my knowledge. Maybe they're using an abacus or something, but that was still more accurate. What they did in the 1850s up to 1970, the reaction time papers, the reaction time data in those studies was more accurate than what most people do now. Because most people in the 1970s, they started using personal computers, and that introduced a crap load of error. Let me show you let me share the screen. And if I can figure out how to share the screen.
It's under share screen.
Yeah, a shared screen. So can you see that?
It says cortical metrics. There we go. Yeah.
Comparison of available technology using robotics.
Yep.
Okay. So anyway, what we're doing is, oh, there you are. Okay, we're on different screens now. But so let's just compare visual reaction time with a touch screen. That's like an iPad. Visual reaction time with USB mouse. That's on a regular laptop or desktop computer, which, you know, delivers a visual reaction time with the first generation brain gauge. That's just a brain gauge like what you have, except you're responding with a computer mouse, and then the Brain Gauge Pro. If you look over on the right, you see the difference in latencies reported. In other words, that is the error. So with the brain gauge, like people in class have, or people that buy brain gauges now, they get a 0.3 millisecond latency. Well, look at the graphs. This is all the data from the study. Interesting, the hostile reviewer was complaining that we didn't show all the data, and we showed all the data. Multiple times we showed all the data. There was nothing to hide because it was collected robotically. But look at this error. Look at how variable this is. This is all over the map. So, and compare it to this line here. But, you know, basically, and here's the other thing. People have written papers about this before. I mean, Bob, you read a lot of these papers by
Plant and so forth, yeah.
Yeah, by Richard Plant, and he was screaming about this in 2007.
Yeah,
yeah. So, so what's really important to understand is people are using this stuff. Dana is a commonly used method. Dana uses an iPad with a mobile device. And actually, it's really hard to find publications with them because they're playing their cards close to the chest. They don't want anybody to see it. And ANAM and Impact basically use this method here. You guys might have heard of Impact because, well, if you did any sports in high school or college, you're required to get baseline testing. Mm-hmm. Now, why does this matter? Why does having a 400 millisecond latency matter? Well,
I know the answer.
Yeah, I want to hear your answer.
Because Mark Towerdall is angry about that?
Yeah.
That's why it matters. Just a guess.
Yeah. So if you have, I mean, let's say even if you at 100 milliseconds, all right, so we found papers that had reaction times recorded of not just like normal reaction times, 200 milliseconds. But we found reaction time papers that said, you know, had reaction time results of 400 milliseconds, 500. We've had a couple that had 1.6 seconds. Okay, Bob, how fast can you react? There you go. That's a second.
Sure. No.
Reaction time. Okay, so the whole thing is this is propagated the concussion mafia. In other words, people that are doing research in the field of concussion, what they say every 5 years is, oh, we just need another 5 years of research to get to the answer. But they're using crap. They're using crap methods.
The funny thing is that the more money you spent on this type of research, the more money we spent as a society, the longer we did this research, the farther and farther away we were actually getting from the answer.
Yeah, and it just keeps getting piled, you know, every firewall that's added and every whatever. And so one computer, so the latency that you see in this right-hand side, those are not set. It's not like you can just subtract that latency and be done with it. Look at the variability that this this stuff introduces. And, you know, it introduces a lot of variability, and that way you can't measure reaction time variability. So right. The variability that you see in something like this, you know, that's okay. Your variability is somewhere between 0 and 20. And then when you're concussed, it pops up to 40. But, you know, if you're going to have an error of 40 to begin with, you really can't measure it. And if you go from one computer to another computer, the latencies you get there are going to be different, and they're going to be different from even within trials.
Right, so, absolutely.
Anyway,
I'd like to throw in my opinion on this. So what Mark is saying is that, you know, the more modern the human interface device that you use, the more modern it is, the more the latency is. And latency, it's what we're really saying is the more you're adding on these random variable time delays that are random. So if you're using a touch screen based device, you know, the latencies can be huge. If you're using just a computer mouse, something simple like that, you're still getting huge, you know, 40 milliseconds when you're looking at, you know, a reaction time that should be, you know, on the order of 200 milliseconds. You're screwing it up right there, you know, by 20% just with a computer mouse. And so, you know, it turns out that the thing that you need to understand is the scientific instrument that you're using. What is it designed to do? It's designed to do a specific thing. And if it's not designed to make a scientific measurement, you either need to make the modifications necessary to do that, or you need to not use that device. Well, modern computers are not designed to be devices for timing human interface reactions. Modern computers are designed to create the illusion that your interface is smooth. It doesn't do this by measuring finer and finer levels of reaction. What it does is it's a combination of how quickly it updates the screen and, you know, where it shoves different bits of its computational overhead and how quickly it updates, you know, input from the human interface device. So to a large extent, the fact that your computer acts smoothly, appears to you to act smoothly, is a deception. And that deception doesn't really show up when, you know, you're you know, writing a Word document or something, but it shows up tremendously when you actually look at the reaction numbers. So your computer is designed to deceive you into thinking that it's operating smoothly, it's interfacing with you smoothly, when it's doing basically just the opposite. But it's sort of like a shell game. It's moving the time delays and everything around to make it appear to be smooth to the user. So it's modern computers are a terrible instrument. And Mark's talking about this in the context of a neurophysiology course, right?
Yeah.
Talking about this, and I'll post this for my students in microcontrollers, because we're going to prove a point. We're going to prove a very simple point, that you can take a $39 microcontroller that's dedicated to taking a measurement like this, and it will be able to measure reaction time. By the end of the term, a bunch of the students are going to have this worked out. They will be able to measure reaction time more accurately, consistently, than anyone running any super fast gaming computer that you can lay your hands on. So any super fast, you know, some of you guys have fast gaming computers. I'm doing my Zoom discussion here on the latest, greatest, fast gaming computer, and it will have much more reaction time variability than a $39 toy, but that's designed specifically by the students during a project to measure reaction time. That's the point, right? But it's not going to be that great. I mean, if you want to do it really well, you got to have an instrument that's really designed for it, like a brain gauge. Well, we will be way down below what you can get with the fastest computer you could lay your hands on. The reason is that the tool is correct for the job, that we're using something that works, you know, for the measurement that we want to make. And, you know, it doesn't have to be a computer. It's like, you know, the big question Mark and I had initially, and turns out, you know, the answer is the same thing. It's like, why is it that you can drop a ruler, or a ruler can drop, you know, on a certain stimulus, like a light will flash and a ruler will drop, and you grab that ruler with your index finger and thumb. And that gives you if you measure the distance the ruler fell, and you calculate back what was the time, the drop time of that ruler, it's more accurate than the typical modern measure of reaction time.
Right. And well, I mean, it all goes back to calibration. And basically we went from the 1850s, 1860s with these German guys using a chronograph and, you know, carefully figuring out the velocity of the chronograph and marking the place on paper, to fast forward. This is only about 10 years ago. One of the papers wrote in the methods section, their entire methods section for reaction time was, reaction time was collected by a personal computer.
Yeah.
Okay, that ain't calibrated, folks. You got to calibrate your measures, and you got to at least have a specification somewhere.
Well, this is something that comes up right now. It comes up across all of medical science, because about 40, 50 years ago, there was a suppression of engineering in medical research. It's like, why pay engineers? You know, you just got to pay the biologist to test a hypothesis. But for about 50 years now, engineers have been seen as, you know, fairly low-level technicians. You know, you guys, you just open a book and you get the answer. But, which is why if you go to most medical research laboratories, they'll have a pretty senior person, you know, running the laboratory, a surgeon or a biochemist, and they'll have a pretty senior technician. They're also sort of a biology-based background usually. And then very frequently, they'll hire an undergrad to do the programming. You know, Mark and I have both been in this position, you know, in the last 30, 40 years. We were like, you know, and you're low person on the totem pole, really. You have very little input into it. All you need to do is make sure that the numbers, you know, flash up on the computer screen or whatever, they get printed out. And that's good enough for the biologist. And the assumption made by many people in medical research is that if a number flashes up somewhere, it has to be right because the machine is fast. And that's absolutely not true.
Yep. All right. So anyway, we're going to scare everybody off with how long this one was. But we're going to make a few more discussions, and hopefully they'll be enlightening, and they'll be about like what impact this. Like, one of the big things about this that we still haven't really gotten through to people, I think, is how many thousands of papers are out there that are just flat wrong.
Well, do you mind if I just
go ahead
and tell people? Okay, when I was a graduate student, when I started as a graduate student, we tested the literature pretty severely in our own very narrow discipline, muscle physiology. We found an error rate of 85%, where they were just simply wrong on the basis of instrumentation. Another guy who is well-known statistician about 10 or 15 years ago published a paper titled, you know, Everything We Know About, you know, Medical Research is Wrong. And his argument was based on statistics and people using the wrong kinds of statistics. And he was up and around the same number, but it was more than half of what was published was wrong. And then recently there have been, and Mark and I will talk about this in a different talk, you know, later on, there have been cases of very modern medical research where they have looked at hundreds of papers and tried to replicate them. And they can only repeat somewhere between 10 and 25%, which means that somewhere between 75 and 90% of everything published in the medical literature is not really accurate enough. It's always precise enough, but it's not really accurate enough to be true. But there's always really, you know, really convincing statistics and precise numbers and tables filled with data and multicolored graphs, but that doesn't make it right. And so it's an actual crisis that's going on in science now called the replication crisis. And, you know, we'll talk about that, I'm sure, in a different talk, but, you know, we've probably said enough for now. And I'm going to post this for students because I'm having you know, in microcontrollers, we're having a project based on making this measurement using what is essentially a toy. And it will be better than anything you can do. It will be about as good as you could do with a wooden ruler, and it will be better than anything you could do with the fastest computer you can lay your hands on.
Yep.
All right.
Cool. Okay. Let's, we're going to stop recording.
Yeah, which is hard to get us to do, but we'll do it.
Yep.